Hi, my name is Merridee Lefner, and I am a PhD candidate in the lab of Dr. Matthew Wanat at the University of Texas at San Antonio. My research project will test whether a brain region called the retrospenial cortex controls changes in reward preference. The Roman poet Propertius coined a phrase that you might recognize: absence makes the heart grow fonder. The meaning behind this saying is that if you go without something you like for a long time, you end up missing it and wanting it even more. Something interesting about this observation, though, is that it indicates to us that our preferences for rewards are not stagnant. They could be changed. I think a lot of us have felt the absence makes the heart grow fonder effect over the past several months of physical and social distancing throughout the global pandemic. When the pandemic first started, we had been spending a lot more time cooking at home because restaurants were closed down. This caused us to miss our favorite restaurants, probably more than we expected. Personally, I really love tacos, but I was not able to go and get them because my favorite restaurant was not open. Does this absence of our favorite foods influence how much we like them? I believe it does. Once the restaurants reopened, I took my first bite of a taco from a local taqueria, and it seemed to taste even better than I remembered. In fact, our research group has recently demonstrated this absence makes the heart grow fonder phenomenon with food rewards. When we made a rodent wait a long time before a reward was delivered, the rodents increased their preference for that reward. The results of our research will tell us more than just why quarantining at home has made us miss our favorite restaurants. Our preferences for food, places, and even people guide the decisions that we make on a daily basis. So, our primary goal is to figure out what brain region is in charge of mediating this change in reward preference. Knowing which parts of the brain control these changes in preference will help us better understand the cognitive processes that drive our choices. One candidate is the retrospenial cortex, or RSC for short. This is a small green area indicated on the screen. Recent discoveries have informed us that the RSC actually receives input from a lot of different brain regions. However, despite this knowledge, the role of the RSC in behavior is largely unknown. We turned off this region in rodents, and it prevented rats from changing their reward preference after waiting a long time. Therefore, we think that one purpose of the RSC is to use our past experiences with rewards to influence our preferences. The preliminary studies we performed can only tell us so much about what the RSC does. What would be really cool is to be able to detect the activity of brain cells in this region and directly manipulate these cells to observe a causal relationship. We are proposing to investigate exactly that with the help of two newly developed techniques for studying the brain. The first technique will require a probe to be inserted into the RSC. This innovative technique will allow me to safely observe and record the activity of a large population of RSC brain cells, all at the same time. With this technique, we are able to see changes in brain cell activity in real time while an animal is waiting for the reward to be delivered. I predict that if we change the rodent's preference by making them wait a long time for the reward, we will see increased RSC brain cell activity to that reward. This would demonstrate a correlation between RSC brain cell activity and a change in reward preference. This correlation would tell us that when our preference changes, so does brain cell activity in the RSC. Even if there is a correlation, that does not mean that changes in RSC brain cell activity cause a change in preference. To examine this, I will use a second technique, which also requires a probe to be inserted into the RSC. However, this technique is different in that it will allow me to actually manipulate brain cell activity by shining a light. This light will turn on or turn off cells within the RSC. I will use this technique to turn on RSC brain cells in response to a specific reward, and I expect that this will make the rodent like that reward more. Alternatively, I will also do the opposite approach and turn off RSC brain cells in response to a specific reward, and I expect that will make the rodent like that reward less. It is important to note that this is a safe manipulation that is not harmful to the animal. Additionally, this is a critical tool because it will take our study beyond correlation. It will demonstrate a causal relationship between the RSC brain cells and changes in reward preference. This is exciting because it will show us that brain cells in the RSC drive changes in preference. Propertius had observed a behavioral phenomenon over 2,000 years ago that still rings true today, but scientists have yet to understand exactly what happens within the brain as preference updates. I believe we are on the right path towards finally understanding which brain region predominantly controls changes in preference. From this study, we will learn two major things. The first is that we will understand the natural activity that goes on in the RSC as our preference for rewards update. The second is that we will learn how direct manipulation of these RSC cells contributes to changes in reward preference. These discoveries will tell us what's been happening in our brains during the pandemic-induced restaurant shutdown and why that long-awaited taco bite tasted better than ever. But on a larger scale, we will learn which part of the brain controls changes in preference, which will help us gain a better understanding of the cognitive processing underlying our conscious decisions. Thank you.